structural and numerical changes of chromosomes. This is also called as the chromosome aberrations, abnormal structures. Okay, abnormal structure and numerical changes of the chromosome is also called as chromosomal aberration. These are the two types of the chromosomal aberrations. One is the structural aberrations, and next one is the numerical aberrations. Mainly structural is the gross structural changes of the chromosomes. Gross structure of the changes of chromosomes. Next one is the numerical aberrations mean polyoid or numerical changes of the chromosome. What are the structural aberrations mean? Especially structural changes of the chromosome involves the gain of or loss of the or reallocation of chromosomes, segments, and genes. In effect, the bring about the changes in the number and arrangement of genetic loci with the marked genetic effects. Structural aberrations are the four basic types, namely deletion, duplication, inversion, and translocation. These are the four of the structural changes, especially structural changes in chromosome mutation, abortion. The first one is the deletion. Okay, deletion. The first one is the deletion. Deletion is the loss of chromosome segment together with the genes contained in it. It is also known as the deficiency. Is also known as deleting. Especially deletion means deletion is the loss of the chromosome segment together with the genes contained in it. It is also known as the deficiency. Loss of the terminal segment with the single break in the chromosome is also called as the terminal deletion. Especially related to the terminal position. Okay, end of the position. Okay, mainly terminal segment with the single break in the chromosome is also called the terminal deletion. Loss of the an intercalary segment with the two break in the chromosomes is also called as intercalary deletions. In any case of the loss, segment would be the eccentric, that is the without centromere. Mainly in case loss of the segment with would be the eccentric, that is the without the centromere. Deletion often results in the abnormal phenotype or the lethality. The deletion is the abnormal phenotype and lethality. See the diagram is the deletions, especially break the some of the chromosomes. Okay, mainly deletion part. Okay, loss of the chromosomes. Okay, next one is the terminal deletion. Especially terminal deletion means the end of the end of the position. Terminal deletions. Okay, in terminal deletion, deficiency with the single break is known as the deletion, which involves the loss of the terminal segment. And next one is the intercalary deletion. Deficiency with the double break is known as the intercalary deletion, which involves the loss of the intercalary segment, loss of the intercalary segment, the terminal deletion, intercalary segment. Types of the deletion. These are the two types of the deletions. Homozygous deletion. A segment is lost from the both the chromosomes homologous pair. It is the mostly used in the lethal. Mainly is that the segment is lost from the chromosomes. Chromosome is the homologous part. Is also called as the homologous deletions. Next one is the heterologous deletions mean a segment is lost from the one of the two homologous uh, one of the two homologous mainly the segment is lost one of the two homologous It mainly causes the abnormal phenotype. Okay, homozygous, heterozygous. Homozygous, a segment is lost from the both the chromosomes homologous pair. It is mostly used in the lethal type. Heterozygous deletion is also a segment is lost from the one of the two two homologous. It is mainly cause the abnormal phenotype. Mainly metatic pairing is homologous with the heterozygous. Deletion involves the formation of an unpaired loop or bluage in the normal chromosome. There, the loop is formed in the region which is the corresponding to the last part of the deficit chromosomes. Genetic effect of the deletions. Deletions involve the loss of the genes. The intensity of their effects depends on the chromosome and quantity of the lost genes. Heterozygous for the small deletions are usually uh, various but the abnormal. However, the homozygous is small deletions may have the lethal effects. Pseudodominancy. Some heterozygous deletions involve the loss of the dominant alleles without affecting their the counterparts in the other homologous recessive alleles due to the deletion of the dominant allele is also called pseudodominancy. Pseudodominance. Example: Waltzing and mouse. It is the most mouse which is moves the about vertically until the gets exhausted is the results of the pseudodominancy. Pseudodominancy. And next one is the human deletion mutations results in the drastic phenotype changes, especially granulocyte leukemia. It is caused due to the deletion in the long arm of chromosome 21. This is the non-irritable. This is the non-irritable mutations occurs when the translocate are too many granulocytes in the blood. It is a condition that is the closely related to chronic myelogenous leukemia, and the other bone marrow is the disorders. Granulocytes are the white blood cells, and the small granules are the particles. Okay, next one is the Cry-Do chart syndrome is also is that comes the deletions. It is well known as the case of the deletion mutations in many. It is also called as the cat cry syndrome, caused by the heterozygous deletions in the they are the cry just like the cat mews. The disorder children is are the mentally retarded, and even they are the cry just like a cat mews. Their disorder is characterized by the intellectual and disability and the delayed the development, small head size, low birth weight, and weak muscle tone, and next one infancy. They also have the distinctive facial features, which is the rounded face. Some children's with the Cry-Do chart syndromes are born with the heart duplicate deletions is over. Duplication is the doubling or repetition of the chromosomes segment during the chromosome duplication. So in this case, a set of gene exists more than once, and this extra set of the generally called repeat. The duplicate segment may be incorporated either with the parent chromosome or with the another chromosome, or it may exist as the separate piece. Duplications are the believed to the important raw material for the organic evolutions. Types of the duplication: random duplication, reverse random duplication, displaced duplication. Okay, random duplication. The extra segment and the parent segment are the each other and both of them have same polarity or order of genes. Reverse tandem duplication, the extra segment lies next to the parent and segment with the reserved polarity or reserved gene sequencing. And if you the displaced duplication, the duplication segment lies some distance average from the parent segment. It gets the translocate of the non-homologous chromosomes. Duplication can take the kinds, namely homozygous duplication and heterozygous duplication. Homozygous duplication, both the homologous of the pair chromosome will have the homologous duplicate sequence. Heterozygous duplication, only one of the pairs chromosomes will have the duplicate sequence. Next one is the tandem duplication and reverse duplication. Reverse mean, for example, AB they can convert the BA. Okay, random tandem duplicate means the AB, CDEF. Okay, reverse. Next one, displaced duplication the same manner, same the chromosomes. Okay, ABC. Next one is the AB, ABEF. The both are the considered displaced. Next one is the displaced duplication is the deficiency chromosomes. Next one, duplicate segment exists the new chromosomes. These are the duplications, deletion and duplication. Genetic effects of the duplication. A classical example tandem duplication is the bar eye of Drosophila. The normal eye of the female Drosophila is oval, whereas the bar eye is like an oblong, the narrow bar. Bar eye is shown due to the duplication of the specific site is the X chromosome is called as the bar locus 16A. 16A, this locus contains the normally narrows the eyes directly. The duplication heterozygous eye bumps in the smaller and the narrow eye that is the bar eye, and the duplication homozygous the eye become the extremely small and double bar eye. Any further duplication of the bar locus would you like make the eyes still the narrower? These are the duplications. Next one is the inversions. Inversion is the reversible of the linear orientation, okay, of chromosome segment and the orders of its gene sequences. Okay. Inversion is the extremely found in the intercalary segments. It involves the breakage of the chromosomal segment and its the subsequent reinsertion into the same locus in the reverse orientation. Normally, two breaks occur in the chromosomes are also called as the inversion points. Types of the breaks, paracentric breaks and next one the pericentric breaks. Mainly paracentric breaks. Paracentric breaks also the break the exclude the centromere and the both the breaks occur on one side of the chromosome centromere in the same arm. The breaks separate an eccentric rotation and the reinsertion and the centric segment is also called as the paracentric inversions. Pericentric, the break include the centromere and they occur on one on other side of the centromere. So anyone in each arm, the breaker separators and the centric arm, and so the rotation and reinsertion of centric segments is also called as the pericentric inversions. Breakdown is the rotation 180 degrees, paracentric. The next one is the pericentric mean maybe changes in different places. Okay, genetic effects of the inversions. The effects of inversions are not also drastic as those the deletion and duplication. One of the best characterized recurrent inversions, the giving rise to disease and the hemophilia and X-linked disorder caused by the mutations, the factors, the eighth recruitment inversion has been the found in the approximately 43% of the patients. Producers and the phenotypic changes due to the change in version serves the crossing over and next one the surprises and reduce the crossing and frequency. Translocation is the another type of the chromosomal evolution, especially the structural changes. Translocation. Translocation is the chromosomal aberration, which is involves the reorganization of chromosomes through the transfer or change in the position of the chromosome segment with the gene or loss of the genes. It involves the transfer of the segment from one location to another, the same chromosome, or from one chromosome to non-homologous chromosomes. Okay, types of the translocation is the intrachromosomal translocation and interchromosomal translocation. Intra, it is the three break in abortions resulting the change of the position of the segment within the chromosome, either from the one or two segments. Interchromosome translocation, it is the transfer of the segment from chromosome to another. It is the two types, transposition and intertransitional translocation. Non-reciprocal one-way transfer of the chromosome segment from one chromosome into another. The reciprocal and mutual interchanges of translocation, mutual exchange of the segments between the two chromosomes, either the homologous chromosomes. Translocational, transchromosomal translocation, and next one is that transchromosomal translocation. Genetic effects of the translocation cause the change of the chromosomes and gene combination, alters the linkage relationship of the gene translocation mainly leads to infertility, miscarriages, and our children's with the abnormalities. Several forms of the cancer caused due to the translocation of the chromosomes and segments, mainly in leukemia. Other than next one is the numerical changes of the abortions. Numerical abortion is also known as the polyid changes. They involve the gain or loss of the either individual or chromosomes or monoploidy chromosomes. The major cause of the polyploidy changes is non-disjunction, and numerical abortions are the are two basic types. One is the euploidy and another one is the aneuploidy. Euploidy is the changes involve the duplication or loss of one or more monocentric chromosomes. Okay, euploidy. Monoploidy, the somatic cells contain the only one base set is the chromosomes. Diploidy, the somatic cells contain the two basic cells set of the chromosomes. Next one is the somatic cells of the contains more than two basic genomes. See it in polyploidy. Next one is the autopolyploidy condition, which is the multiple genomes are the identical and derived from the same species. Allopolyploidy condition, which is the multiple genomes are the distinctly and different and the derived from the different species chromosomes through the hybridization. Next one is significance of the polyploidy. Polyploidy serves as the mechanism of the specification and evolution. Any polyploid is the found in many plants, and allopolyploid is the many from the many hybrid, such as the mule. They are the significant in agriculture and the horticulture. Aneuploidy is the condition which is the individual chromosomes of the chromosomes. Chromosomes are the pairs and added and lost from the somatic chromosome complement. And it is usually results from the mitotic or metatic non-dissection of the aneuploidy is the two kinds. Haplopolyploidy is the aneuploidy due to the chromosome loss. The monosomy, one chromosome is lost from the diploid set of the chromosome. Example, turnover syndrome. Next one is the nullisomy, loss of the homologous pairs chromosomes from the somatic set 2n minus 2. These are the don't they often don't survive. Hyperpolyploidy, it is the aneuploidy due to the chromosomes gain. Hyperpolyploidy, it is the aneuploidy due to the polyploidy. Trisomy, addition of the extra chromosome and diploids to n plus 1. Down syndrome, trisomy. Edwards syndrome, trisomy 18. Potato syndrome, trisomy 13. And Klinefelter syndrome, the XXY is another X of chromosomes. Klinefelter tetrasomy, addition of the homologous part of the chromosome diploid to the tetrasomy. Example, the polycystic Klein syndrome and tetrasomy. And next one is the cat eye syndrome, the tetrasomy 22. Okay, these are the chromosomes. Okay, mutations. Next topic is the mutations. Mutations is the mainly is the mutation is change in occurs in our DNA or sequence either due to the mistakes when DNA is the copied or as the results environmental factors which is the UV light. The DNA sequence of genomes can be altered in the number of ways. The gene mutations have the varying effects on the health depending on where they are occurred in the whether they are altered the functions in the essential proteins. And types of the mutation, gene mutations are the two types. And gene mutations, this one is the types of gene mutation, the chromosomal mutation. Gene mutations also they can divide the point mutations and frame shift mutations. Point mutations, especially silent and missense and nonsense mutations. Frame shift mutation, addition and deletion. Point mutations. A point mutation or substitution is the genetic material where the single nucleotide base is changed, insert or deleted from the sequence of DNA or RNA. Point mutations. Mainly, point mutation is the silent and nonsense missense mutation. Silent mutations are the mutations in DNA that do not have any observable effect on the organisms in the phenotype. There are a specific types of the neutral mutations, nonsense mutations. A nonsense mutation also change in the DNA base pair. Instead of the substituting one amino acid for the another one, however, alter the DNA sequence is the prematurely signals the cell to stop the building a protein. This type of the mutation results in the shortened and the protein that may functions improperly or not to all. Missense mutation. This type of the mutation is a change in one DNA base pair that is the results in the substitution of the one amino acid for another in the protein made by the gene. Frame shift mutation. This type of the mutations occurs when the addition or loss of the DNA loss of the DNA base changes in gene reading the frame shift. A reading frame consists of the group 3 base pairs, which is the code for one amino acid. A frame shift mutation shifts the grouping of these bases and changes the code for the amino acid. The resulting protein is usually non-functional. Insertion, deletions, and duplication can be also the frame shift mutations. Deletion. A deletion changes the number of DNA bases by the removing the piece of DNA. A small deletions may remove one or few base pairs within the gene, while larger deletions can be removed and the entire gene or several neighboring genes. The deleted DNA may alter the functions of the resulting proteins. Insertion. An insertion changes the number of DNA bases in gene by adding the piece of the DNA. As a result, the protein made by the gene may not function of the properly. Chromosomal mutation, gene and deletion and duplication, inversion and translocation. Chromosome mutation, a chromosome mutation is an the un, unpredictable changes that occurs in the chromosome. The changes are the most often brought on by the problems that occur during the meiosis and by the mutagens and chemical and radiation extra. Deletion, a small scale type of the deletion, multicolor and is one of the which is one or more nucleotides are lost or deleted from the chromosomes. Duplication, a type of the mutations in which is the proteins inversion mutations, translocation mutations. Okay. The next one is the mutation and its detections. Mainly, what are the characteristics of the mutations is the mostly it is the mostly recessive and the very rarely dominant. Now, most of the mutations have the harmful effects and the very few of them 0.1% the beneficial. Mutations may be due to the change in gene, a group of the genes in entire chromosome. And mutations may be due to the change gene, a group of the genes or the entire chromosome. If gene mutations are not lethal, or the mutations individually may survive. If mutations occur the both locate and simultaneously, the mutants can be identified one generation. However, it is the restricted to one locus only. Dominant the effect of this been the M2 generation. What are the characteristics of the mutation? Many of the mutations show the sterility. Most mutants are the negative selection value. Mutations are the random. They are can be occur in the any tissue cells of an organism. Mutations are recurrent. That is, the same mutation may occur again and again. Induced mutations commonly show the pleiotropy, often the mutation closely linked to the genes. Already we discussed the types of the mutations, spontaneous mutation. Based on the source induced mutations, the mutation that originates the response to the mutant treatment, for example, crop and mutant varieties and original varieties and mutagens, mainly rice. Jagannath Masuri mutant is the original variety is the T141, mainly mutagen is the X-rays and gamma rays. Next one is the based on the direction two type forward mutation and reverse mutation. Any change from the wild type into the mutant allele, reverse mutation change from the allele in the wild type. This is based on the site nuclear mutation and cytoplasmic mutation. Based on tissues, germinal mutations, the gene is altered in the germ cells because of the germ cells give rise to the gametes, somatic gametes, while carry the mutation and there will be a possible generation based on tissue somatic mutations. Based on character morphological alerts, the morphological character also known as the visible mutation, dwarf fish, short-legged sheep in the curly wings in the Drosophila. Biochemical mutations, this is the uh, alters production of the biochemicals by the organism based on the visibility. Macromutants in the micromutants, mutations with a large number of changes in the phenotypes, the macromutant, micro mutations involved in the phenotypic changes. Next one is the easily detected in the macro mutations, not easily detected in the micromutants. Found in the qualitative characters in macromutants, observed in the quantitative characters in the micromutations. The based on the survival lethal mutation kill the all the individual lethal mutations. Sublethal kill the most of the individuals. Subvital mutation kill the same of the individuals. Vital mutation Vital mutation don't affect of the survival. So super vital mutation increase the survival based on the cytology chromosomal mutations, changes in the either chromosome stem structure, gene mutation, alteration based sequence, concerned genes and changes. Based on the effect on the functions, loss of the function mutation, reduced function, gene of the function mutation, mutants, the morphological mutants detected by the onwards appearance of the individual shape, size, and the color. Lethal mutagens, especially physical mutagens and chemical mutagens, include the various types of the radiation is the physical mutagen, X-ray, gamma rays, alpha particles, beta particles, fast and the thermal neutrons, UV rays. The chemical mutagens, alkylating agents, base analogous, acridinides, and other mutagens, and nitrous oxide hydrothermal. These are the chemical mutagens. This is the materials used for the treating of the mutagens. Seeds and pollens, and next one is the whole plants, ba- vegetable buds, and the bulbous tuber stickers. What are the significance in the plant breeding in mutations? And desirable character linked with the undesirable character, to know the source of the resistant gene in the available germplasm, to create variability, to develop the male sterile lines, to create variation vegetatively propagated plants. And next one, how to detect the mutation, detection of the mutation. Scoring of the some types of the mutation in certain organisms is relatively easy. For example, mutations and an animal and resistance in bacteria are simply detected in the plant bacterial cells on a medium of containing and normal concentration of the concerned antibiotic selective medium. The colonies they develop in such medium will be produced by the cells resistant to the antibiotic. But detection of morphological mutations in eukaryotes requires the examination each individual of the population for the mutant phenotype. This is not only tedious, requiring must be the source of the detection of the mutations in the prokaryotes, detection of the auxotrophy mutations. Auxotrope mutants are the unable to synthesize biochemical essential for their growth and the development. Wild type cells are the capable of producing. Therefore, therefore such mutations cannot be detected using the simple approach described the before their mutants detected by the replica plating. The replica plating is the technique is developed by the Joseph Esther Ledenberg in 1952, direct selection of the bacterial mutations. Next one is the master plate. In this technique, all cells are the plated into the complete medium inoculation obtain the distinct colonies. It is also called as the master plate. And block of food is our cork of the size suitable of the master plate is the covered with the valvulate cloth. This block is the sterilized and lowered in the master plate till the relevant touches all the colonies. Now the block of the withdrawn and the gently lowered onto the plate containing the selection media, so that is the bacterial cells sticking of the plates replicate. Point marked both on the master plate is the replicate the this makes it the possible locate in the master plate colony. The colonies that they develop in the selection of the media plate is the wild types of the cells. Next one, the AMIS test is also, this test was developed by the Bruce Amis and the co-workers and based on the study requiring auxotrope mutants in the Salmonella typhonurium. The routine arms and test address both these the needs as the follows. The cells are the plated onto the media that contain the trace insulin, which is enough all the few cells divisions and the inadequate usable colony formation. Next one is the C-based stock. Next one is the C-based stock of Drosophila. The another method involves the C-based stock method is carries in inversion and the heterozygous state of workers the cross over the suppressor and recessive lethal on X chromosomes in heterozygous state and dominate the marker and buried thread. One of the two X chromosomes in female fly carried in all these three features, and this is the other X chromosome was the normal male flies. And eradicated for the induction of the mutations were crossed in the CBA females. Male progeny receives the CIB X chromosome wild type, CIB method. An attached X chromosome technique. This technique also based on the attached X females designed the study of the visible sex-linked mutations in the Drosophila mutagen and treated the males are the mutated with the attached X females. The X superior female in the YY progeny produced from the such crosses due to the survive only the XY progeny is recovered. All the male XY progeny and receive their the Y chromosome from the attached X female parent, while there the X chromosome is the contributed by the mutagens treated the male parent. The Russell test in dominant lethals. The dominant lethal assay is the mice is initially is the Russell test, and co-workers subsequently extend the study of the mutagenity checks, and therefore the system of in the Russell test is dominant and detection of the mutation in plants. This technique detection of the mutation relatively poorly develops the plant species. The following three approaches adopted for the purpose. In some species of maize, in the strains homozygous for the several recessive genes as well as those for the dominant alleles of the genes are the available. In such case, the seeds or plants strain the homozygous for the several dominant genes treated with the mutagen. These plants also are crossed with the strain having the recessive alleles for the same gene in homozygous state. The treated in the plants are the generally used in the female parent due to the partial male sterility in the mutagen treated in plants in the progeny of the cross plants exhibiting the recessive forms and the conserved characters are the compound. The plants showing the recessive from the trait will recessive one recessive allele from the tester parent and next having the recessive traits, but the other recessive alleles would have been the produced due to the mutation of the mutagen treated parent and is the parent and the dominant allele of the genes. The most of the plant species, our such strains are not readily available in the species, seeds and the variably. The above produced applicable mutations are the detected quantity and the traits of the macronutrients, macro mutations. Okay, these are the detection mainly CIB method in Drosophila, CIB method according to our syllabus. Molecular based gene mutations is that the gene mutations also substitution mutation and frame shift mutation. Substitution mutation is the transition and transversions. Frame shift mutation, deletion, mutation, incident, and ionization, depurinization mutation, the base analogous, and deamination transition. Spontaneous gene mutations mainly cause of the spontaneous mutation. The spontaneous is the genetic change that occurs naturally without any external influence, such as the radiation or chemicals. These mutations rise due to the errors in DNA replication, spontaneous chemical changes in DNA bases, the action on the mobile genetic elements, transposons, cause of the spontaneous mutations, errors in DNA replication, mistakes by the DNA polymerase that is the escape the proofreading, tautomeric shifts, rare forms of the DNA base cause the mispairing during the replication, depurination and the deamination, loss of the purine A and G are the conversion and the cysteine. Next one is the oxidative damage. Reactive oxygen species ROS can damage DNA causing the base modification. Transposon jumping genes, DNA sequence that is the moved within the gene disturbing the normal gene function. Types of the spontaneous mutations, point mutation, frame shift mutation, repeat exon. What are the biological impact of the spontaneous neutral, no effect on the organism. Beneficial, provides the advantage antibiotic resistance bacteria. Harmful, causes the genetic disorders or cancer, sickle cell anemia. Next one is the physical and chemical mutation, chemical mutagens. And the physical and chemical mutagens are the agent that is the cause the mutations by altering the genetic mutations of an organism. They can be classified. Physical mutagens, these are the mutagens cause DNA damage through the physical means, primarily radiation, ionization radiation. These are the high energy radiation penetrate the cells and cause the DNA breaks. Example, X-rays, gamma rays, and cosmic rays. Effects on the double-stranded DNA breaks the chromosomal rearrangements. Point mutation due to the free radical formation. Non-ionization radiation. These are the radiations have the So this is the mutations and chromosomal abortion. These are the two topics are over today. Thank you.